Hi everyone, welcome to chapter 4. We're going to be talking about the plasma membrane in this chapter and all of its wonderful functions. So when we look at the plasma membrane, remember that it consists of a double phospholipid bilayer. And so remember that looks like this. We have little tiny phospholipids heads with the tails. And then we have little heads here with the tails sticking up. So it's a bilayer with the hydrophilic heads up here and the hydrophobic tails here. So what does hydrophilic mean? Hydrophilic means water loving. And so the heads will enter into fluid and will be able to work within that fluid. They can dissolve within that fluid. Whereas hydrophobic tails, this region right here, these are water hating. Another thing you can think of when you think of hydrophilic, water loving, these are polar regions. And then these are the non-polar. So within the plasma membrane, we also have multiple proteins that have many different functions. And we have special molecules called cholesterol, or another form of molecule like cholesterol, maybe ergosterol is an example, that stabilize the plasma membrane. So cholesterol is found in animal cells. Ergosterol would be found in fungal cells. So what is the major function of the plasma membrane? The major job is right here. It helps to regulate what enters and exits the entire cell. So it's basically like the bouncer at a bar. It's going to regulate what comes into and what leaves that cell. So the plasma membrane, I said, had different proteins with different functions. One type of protein that the plasma membrane has is called a peripheral protein. Peripheral proteins are going to be proteins that are found on one side or on the other side of the plasma membrane. So they're going to either be found inside or on the outside of the membrane, but they don't span all the way through. Whereas integral proteins are found spanned all the way through. So they span the entire membrane. So if I were to draw this, again, you have your little plasma membrane phospholipids here. I'm going to draw a couple extra just so I can draw these proteins too. And I'm just putting a bunch of tails in between, okay? I'm going to change the color just so you can see what the protein looks like, and I'll just use a darker color for the protein. A peripheral protein would sit on the ends like that, okay? Whereas an integral protein is going to move all the way through. So it's going to be found spanning all the way through, going across. That's the major difference between these two types of proteins structurally, and they have different functions as well. So peripheral proteins may be proteins that have enzymatic activities, so they might catalyze different reactions, or they might help in identifying structures, whereas integral proteins, I already mentioned, they protrude the entire membrane, so they can be seen on both sides, and they're embedded, and they can move within the membrane from one area to another, but they can't be removed very easily, whereas the peripherals can be removed easily. And so this brings us to the fluid mosaic model. A fluid mosaic model is how that plasma membrane looks and how it stays together. So it's not a static structure. it is very mobile or fluid. So it's constantly changing in its shape. So let's look at that. Hold on 1 second. In 1 second, we'll look at that. So first, we'll talk about glycolipids and glycoproteins. So there are structures within the plasma membrane that have carbohydrates attached. A glycolipid would be a lipid that has a carbohydrate group attached. So you might have that same plasma membrane. I've just changed the color because I already changed the color of the pen. Okay, and then you might have, and I'm just going to put this as a glycoprotein. Okay, so there's your protein. And then you might have a carbohydrate attached right here to this lipid, and that would be called a glycolipid. Or you might have a carbohydrate attached here to this protein, and that would be called a glycoprotein. Okay, and so now, as I said before, let's look at what that membrane looks like. But boom, right? Here it is. So here we have that integral protein that spans across the entire membrane here. And over here you can see a peripheral protein. Here's another peripheral protein. Here's another peripheral protein. You have carbohydrate, or not carbohydrate, I'm sorry, you have cholesterol here, you have cholesterol here, there's cholesterol right here. Up here we have carbohydrate groups attached to phospholipids, those are glycolipids. And here we have a carbohydrate group attached to a protein, so that would be a glycoprotein right here. So we have all these structures embedded, and you can see that it's very fluid, so it can move. These proteins can move within the plasma membrane. The cholesterol functions in helping to maintain the structure. So it actually helps to strengthen that plasma membrane. And then I'll talk about the different functions of the proteins in just a moment, okay? So there are 5 major types of proteins that we talk about, or 5 functions of proteins. I shouldn't say 5 major types because there's a lot of types of proteins, but within the plasma membrane, there are 5 functional groups of proteins. We have proteins that span, so these are integral proteins, and I'm going to write that up here. And their job is to allow materials to move across the membrane. So remember, the plasma membrane is selectively permeable. It's kind of like a bouncer. It's what's going to determine what goes into and out of the cell. So channel proteins are one way that molecules can move across the plasma membrane, either going into or out of a cell. We also have carrier proteins, another integral protein. So this is also integral. This one's integral. Carrier proteins carry molecules across the plasma membrane into or out of the cell. We have cell recognition proteins. So these are typically glycoproteins, are cell recognition proteins. So these are kind of like your identification tags. This is your ID. And so your cells have your own glycoproteins where other cells have different glycoproteins. And so your body knows the difference between your glycoproteins and foreign glycoproteins. And this actually helps us in maintaining health of the entire body. Then we have receptor proteins. Receptor proteins are proteins that allow molecules to bind, and they might change different activities within the cell. So an example of a receptor protein would be a receptor that binds human growth hormone. And then when human growth hormone binds, it changes the activity of that cell, telling that cell to start replicating, undergoing mitosis. That might be an example of what a receptor protein might do. So it just binds molecules, and when the molecule binds, it changes the activity of the cell. And lastly, we have enzymatic proteins. So enzymes are molecules that speed up a reaction, and they do it by decreasing the amount of energy it takes for the reaction to occur. So enzymes are catalysts that speed up reactions, and I'm going to write that here. Up reactions. And enzymatic proteins, this one is shown as an integral protein, but enzymatic proteins can be proteins that are peripheral. They might be found on one side or the other. Receptor proteins can also be peripheral proteins. Okay. I'm going to actually stop here because this has been long enough, and we'll get into our next video shortly. Bye.